Welcome to another video. This one is microbial growth. So let's microbiology it. Don't forget to like, comment, share, and subscribe. Psychrophiles are microorganisms that thrive in extremely cold environments, typically between negative 20 degrees to 10 degrees Celsius. These cold loving organisms are commonly found in icy habitats such as glaciers, deep ocean waters, Arctic and Antarctic regions, and even permafrost. Psychrophiles have adapted to survive these harsh conditions through specialized cellular structures, enzymes, and metabolic processes that remain active at low temperatures. Their cell membranes are rich in unsaturated fatty acids, which keep them flexible, while their enzymes are specially adapted to function efficiently in the cold, preventing them from freezing. Psychrophiles are of great interest in biotechnology for their potential use in cold temperature applications such as bioremediation in cold environments and the production of cold active enzymes for industrial processes. Mesophiles are microorganisms that thrive in moderate temperatures ranging from between 20 degrees Celsius to 45 degrees Celsius. They are the most common type of microbes found in diverse environments, including soil, water, and the human body, where they play critical roles in processes like decomposition, fermentation, and even the human health. Most pathogenic bacteria that infect humans, such as E. coli and Staphylococcus aureus, are mesophiles because they are well adapted to the human body temperature of 37 degrees Celsius. Mesophiles are essential in industries like food production, where they are used in fermentation to make products like yogurt, cheese, and beer. Understanding mesophiles is crucial in microbiology because of their impact on both beneficial applications and potential health risks. Thermophiles are a fascinating group of microorganisms that thrive in extremely hot environments, typically between 45 degrees and 80 degrees Celsius, such as hot springs, hydrothermal vents, and volcanic areas. These heat loving microorganisms have adapted to these harsh conditions by developing specialized proteins and enzymes that remain stable and functional at high temperatures, unlike those of most organisms. One of the most well known thermophiles is Thermus aquaticus, which produces the enzyme Taq polymerase, a critical component in the PCR technique used in molecular biology. Studying thermophiles not only helps us understand the limits of life on Earth, but it also has practical applications in biotechnology, industry, and even the search for life on other planets. Acidophiles are fascinating microorganisms that thrive in extremely acidic environments, typically with a pH level below 3. These resilient organisms can be found in places like volcanic soil, hot springs, acidic mines, drainage, where most life would really struggle to survive. Acidophiles have adapted to these harsh conditions through specialized cellular mechanisms such as protein pumps that actively expel excess hydrogen ions to maintain their internal pH, and unique enzymes that remain stable and functional in acidic surroundings. Studying acidophiles not only helps to understand the limits of life on Earth, but also has practical applications in biotechnology, including bioleaching for metal recovery and the development of acid stable enzymes for industrial processes. Yeast belong to the fungi kingdom and are best known for their role in fermentation and baking. These single celled organisms convert sugars into carbon dioxide and alcohol, a process that not only helps bread rise, but also plays a critical role in producing alcoholic beverages like beer and wine. At the end of the lesson, I will share questions from my new book called Microbiology It Questions and Answers. Now, back to the lesson. Mold is a type of fungus that thrives in damp, warm, and humid environments, making it a common household problem, especially in areas like bathrooms, basements, and kitchens. It reproduces through a tiny, lightweight spores that travel through the air and can quickly take root on any surface where moisture is present. While some molds are relatively harmless, others can produce allergens, irritants, and even toxic substances known as mycotoxins, which can cause health issues such as respiratory problems, allergic reactions, and skin irritations. Preventing mold growth involves controlling moisture levels through proper ventilation, fixing leaks, and using dehumidifiers in high humidity areas. If mold is already present, well, it's important to address it promptly to maintain a healthy living environment. Isotonic solutions are those that they have the same concentration of solutes as the fluid inside our cells, maintaining a balanced state where water moves in and out of the cells at an equal rate. This balance is crucial for maintaining normal cell function and is often emphasized in medical treatments such as intravenous fluids to ensure that cells do not shrink or swell. Isotonic solutions like normal saline are commonly used in healthcare settings to hydrate patients, replace lost fluids, and maintain blood pressure without disturbing the body's natural fluid balance. Understanding isotonicity is key in both medical and exercise contexts, as it helps maintain the body's homeostasis during various activities. In hypertonic solution, the concentration of solutes outside the cell is higher than inside the cell, causing the water to move out of the cell through osmosis. This movement of water leads to cell shrinkage or crenation in animal cells as the cell loses water and volume. Hypertonic environments can severely impact cell function and survival as the loss of water disrupts essential cellular processes. Understanding hypertonic solutions is crucial in various fields from biology to medicine because it explains how cells react in their environment and why maintaining the right balance of solutions is vital for the cell health. In hypotonic solution, the concentration of the solutions outside the cell is lower than inside the cell, leading to a higher concentration of water outside. Now, this causes water to move into the cell through osmosis, making the cell swell, and in some cases, even burst. Hypotonic environments can significantly impact cells, particularly animal cells, as they lack a rigid cell wall and counteract the influx of water. Understanding hypotonic solutions is crucial, especially in medical and biological contexts, as it highlights how cells respond to different fluid balances, which is vital for maintaining cellular health and function. Obligate aerobes are microorganisms that require oxygen to survive and grow because they rely on aerobic respiration to produce energy. These organisms use oxygen as the final electron acceptor in their electron transport chain, allowing them to generate ATP efficiently. Obligate aerobes thrive in oxygen rich environments such as the human respiratory tract, soil, and water surfaces. Common examples include Mycobacterium tuberculosis, the bacterium responsible for tuberculosis, and Pseudomonas aeruginosa, often found in soil and water. Due to their dependency on oxygen, obligate aerobes cannot grow in anaerobic conditions, making oxygen an essential factor for their survival. Obligate anaerobes are microorganisms that thrive in environments completely devoid of oxygen. Unlike aerobic organisms, obligate anaerobes cannot survive or grow in the presence of oxygen, as it is toxic to them. This is due to their lack of enzymes like catalase and superoxide dismutase, which protect against reactive oxygen species. Now, these microbes obtain their energy through anaerobic respiration or fermentation, relying on an alternative electron acceptor such as nitrates or sulfates or carbon dioxide instead of oxygen. Common examples of obligate anaerobes include Clostridium, Bacteroides, and methanogens, which are often found in environments like deep soil layers, the human gut, and marine sediments. Understanding obligate anaerobes is crucial, especially in clinical settings, as some species can cause serious infections when they grow in oxygen deprived areas of the body. Facultative anaerobes are versatile microorganisms that can thrive in both oxygen rich and oxygen poor environments. Unlike obligate anaerobes, which can only survive in the absence of oxygen, facultative anaerobes can switch their metabolic processes based on the availability of oxygen. When oxygen is present, they use aerobic respiration to generate energy efficiently. However, in the absence of oxygen, they can switch to anaerobic respiration or fermentation to continue producing energy, although less efficient. This adaptability allows facultative anaerobes to inhabit diverse environments from human tissue to soil and water, making them important layers or players in various ecological and industrial processes. Examples include Escherichia coli and Staphylococcus aureus, which can be found in both oxygenated and anaerobic conditions. Aerotolerant anaerobes are a unique group of microorganisms that thrive in environments without oxygen, but can tolerate its presence without being harmed. Unlike strict anaerobes, which are damaged or killed by oxygen, aerotolerant anaerobes do not use oxygen for energy production and rely on fermentation to meet their metabolic needs. They they process enzymes like superoxide dismutase and peroxidase, which help neutralize toxic oxygen species, allowing them to survive in oxygenated environments. Common examples include species like Lactobacillus and Streptococcus, which are often found in the human body and play essential roles in processes like fermentation and gut health. Understanding these microbes is crucial in microbiology, especially in food production and medical contexts where their resilience in oxygen to oxygen is beneficial. Microaerophiles are a unique group of microorganisms that require oxygen to survive, but in much lower concentrations than what's found in the atmosphere. They thrive in environments with only about 2 to 10% oxygen, compared to the 21% found in normal air. This delicate balance is crucial for their survival because higher oxygen levels can be toxic to them. Microaerophiles are commonly found in environments like the human stomach, deep in soil, or in aquatic sediments where oxygen is limited. A well known example is Heliobacter pylori, a bacterium that can cause stomach ulcers. Understanding microaerophiles helps us appreciate how diverse and adaptable microbial life can be, thriving in niches that would be hostile to other organisms. Nitrogen is a fundamental component of amino acids, the building blocks of proteins, making it essential for protein synthesis. During this process, nitrogen is incorporated into amino acids, which are then assembled into proteins according to the genetic code. Without nitrogen, cells cannot produce the proteins needed for structure, function, and regulation within the organism. Sulfur also plays a vital role in protein synthesis as it is a key component of the amino acids cysteine and methionine, which are essential building blocks of proteins. Cysteine contributes to the formation of disulfide bonds, crucial for stabilizing protein structure, while methionine often serves as the starting amino acid in protein synthesis. These sulfur containing amino acids are fundamental to the proper folding and function of proteins in microbial and cellular processes. Carbon is a fundamental element in microbiology, serving as the backbone of all life forms. It plays a very important role in the structure of biomolecules like carbohydrates, proteins, lipids, and nucleic acids, which are essential for growth, reproduction, and functioning of microorganisms. Microbes participate in the global carbon cycle by breaking down organic matter, releasing carbon dioxide, and converting it into biomass through processes like photosynthesis and chemosynthesis. Understanding carbon's role helps us grasp how microbes impact ecosystems, influence climate change, and contribute to essential processes like fermentation and biodegradation. Phosphorus is a key component of the phosphate backbone in DNA and RNA, linking nucleotides together to form the structural framework of these molecules. It helps create a strong phosphodiester bond between the sugar molecules of adjacent nucleotides, providing stability in the double helix of DNA and the single stranded structure of RNA. This essential role of phosphorus is very important for the storage and the transmission of genetic information. Nitrogen is a key component of nucleotides, the building block of DNA and RNA, forming the nitrogenous bases adenine, guanine, cytosine, thymine, and uracil that encode genetic information. These nitrogenous base pairs specifically to create the double helix structure of DNA and a single stranded structure of RNA. Nitrogen's presence in these bases is essential for proper genetic replication, transcription, and protein synthesis. Trace elements. Trace elements are essential micronutrients required by microorganisms in minute amounts to support vital cellular functions. In microbiology, elements like iron, zinc, copper, manganese, and molybdenum play crucial roles in enzyme activity, DNA synthesis, and maintaining overall cellular structure and metabolism. Although needed in small quantities, their presence significantly impacts microbial growth, survival, and pathogenicity. For example, iron is a key component of cytochromes involved in electron transport and energy production, while zinc is very important for stabilizing enzyme structures. Understanding the role of trace elements helps microbiologists better grasp how microbes thrive in various environments and adapt to nutrient limitations. Organic growth factors. Organic growth factors are essential compounds that microorganisms cannot synthesize on their own, such as vitamins, amino acids, and nucleotides. These factors are vital for microbial growth and metabolism as they serve as coenzymes, precursors, or building blocks in cellular processes. Microbes must obtain these nutrients from their environment to thrive, influencing their ability to survive in various ecological niches. Unlock your microbiology knowledge with my comprehensive Microbiology It questions and answers book designed to help you ace your exams and retain what you study. With over 450 questions and answers covering key topics, it's the perfect study companion for you. Want to dive deeper? Well, click the link in the description to get your copy today and take your learning to the next level. Well, next up are my chapter interactive slides for my questions and answers. Click the link below to get yours today. Microbial growth. What are the physical requirements for microbial growth? Name the primary microbial groups and list their optimal temperature ranges. What is the name of Archaea's microbial group and what is its optimal temperature? What is the pH optimum growth for microbes? What are acidophiles and what is their pH optimum growth? pH 5 to pH 6 is the optimum growth for what microorganism? Can alkalinity inhibit growth? What is used as a pH neutralizer to allow growth? Name the two specific neutralizers. What type of osmotic environment are microbial cells in when water has to leave the cell, and what is this process called? What can grow in high salt concentrations? Obligate halophiles can grow in which sea? Which halophile can grow in 2% salt concentration? What type of osmotic environment are microbial cells in when water enters the cell and causes it to swell? What are the chemical requirements for microbial growth? Well, thanks for watching my video. Don't forget to like, comment, share, and subscribe. Well, I'll see you in the next one.